Hello guys, welcome to Dr. Science. So today we are going to talk about how insulin is secreted from the beta cells of pancreas. As you can see, this is a complex diagram taken from the internal medicine. So I will come to this later. First I will explain to you guys, okay? So suppose here is a big cell, and this big cell is called as the beta cells of pancreas. Okay? So these cells are containing a special type of transporter. As you can see here, there is a special type of transporter. Okay, and what is the function of this transporter? It will transport the glucose. Here is your glucose molecule. It is transferring these glucose molecules into your cells. Hence they are called as glucose transporters. Okay, or we can simply call them as GLUT. Okay, glut, or we call as glut transporters. GLU stands for glucose, T transport transporter, and mainly it is type 2. Actually gluts are mainly divided into certain types based on the organ systems they are present. On beta cells of pancreas it is glut 2. Okay, so once the glucose comes in, what will happen? This glucose is getting converted into pyruvate. Okay, I will draw this step here so you will understand. So what happens once the glucose enters the beta cells of pancreas? Glucose is getting converted into pyruvate. And what is this? If you know biochemistry, glucose getting converted into pyruvate is called as glycolysis, right? And during this step, what is formed? During this step, ADP is converted into ATP, right? So here what will happen? ADP is getting converted into ATP, right? So one of the function of this ATP is there are certain types of channels here, and these channels are called as ATP sensitive potassium channels. Okay, the channels here are called as ATP sensitive potassium channels, because this ATP, as you can see here, it is coming and inhibiting this potassium channel. Okay, that's why they are called as ATP sensitive, because when ATP is more in the cell, it will inhibit this potassium channel. So once they inhibit this ATP sensitive potassium channel, potassium cannot escape outside, right? And potassium gets accumulated in the cells, lots of potassium. So as you know from physiology, normally in the cell it is negative. When the potassium get accumulated in the cell, it moves from negative to positive, and it is called as depolarization. Because potassium, there is lots of potassium getting accumulated in the cell, and these positive cations will cause depolarization, right? And this depolarization produces a current. A current is produced in the cell, or a voltage is produced. And this voltage, what it will do? It will activate the calcium channels. This voltage will activate the voltage gated calcium channels. Okay, what will happen when they are activated? This voltage gated calcium channels, when they are activated, calcium goes inside. This is very important step. Okay, once the calcium goes inside, what will happen? I will just draw it here because the space is not enough there. So let me draw it clearly. So these are the beta cells of pancreas. So there is certain type of voltage here, and the voltage gated calcium channels are activated, right? So as you can see, the calcium is getting inside. Let me draw it clearly. So here are the voltage sensitive calcium channels. So calcium is getting inside. So once the calcium gets inside, what will happen? When the calcium gets inside, it will activate cyclic AMP. Okay, me draw it clearly here. The calcium will go and increase the cyclic AMP levels, right? So what will, when this cyclic AMP is activated, it will release secretory granules which contains insulin. As you can see, there are granules here which contains insulin inside it. Insulin is present in these secretory granules. So this increase in CAMP, cyclic AMP, will release this secretory granules and insulin is released outside. Okay, so that was about the insulin secretion. Now we will take a look at the diagram. So as you can see, what will happen, these are blood glucose transporters which are present on the beta cells of pancreas, right? Mainly type 2, blood type 2. So as you can see, glucose is getting inside and glucose is getting converted into pyruvate, during which ATP is produced. ADP is converted into ATP. So this ATP will inhibit this potassium channel, right? So there is no more potassium efflux and there is increased potassium inside the cell, right? And this increased potassium will cause depolarization. Can you name a drug which can inhibit this ATP sensitive potassium channel? Yes, you are right. It is sulfonyl ureas. And the other things are glinides. Okay, glinides and sulfonyl ureas can inhibit this ATP sensitive potassium channel, and thus insulin will not get released. Okay, so as you can see, once depolarization or voltage is produced, the voltage dependent calcium channels are opened and calcium is getting inside the cell. Once calcium is getting inside the cells, it will increase the cyclic AMP levels, and this cyclic AMP levels, increase in cyclic AMP levels, will release the beta insulin to the outside. Along with insulin, you will also see C peptide and IAPP, okay, which is also, this is IAPP is also called as amylin, which are used for diagnosis of diabetes mellitus. Okay, all these three. So that was about the how insulin is secreted. Thank you.